I'm Jishnu Das. I'm an assistant professor at the Center for Systems Immunology at the University of Pittsburgh. And I'm going to be talking to you about a project that my lab recently completed, where we used a network-based approach to identify expression modules underlying rejection in pediatric liver transplantation. And this work from my lab was recently published earlier this year in the journal Cell Reports Medicine. So we wanted to predict liver transplant outcome using an interpretable machine learning approach. So kids, for a variety of reasons, ranging from biliatresia to hepatoblastoma to other metabolic disorders, need liver transplants. However, unfortunately, even after getting a transplant, a significant number of them suffer from what is known as acute cellular rejection. So they reject the liver that was transplanted. This is different, discrete from rejection in other solid organ transplantation contexts, which are often antibody mediated. So for example, in the context of kidney transplantation, you have antibody mediated rejection. And there are much better criteria, for example, HLA cross matches to predict whether someone will experience rejection or not. So these can be used for screening upstream, but no such criteria exist in the context of pediatric liver transplantation, which is why this is an underserved area, and we need signatures that can predict the risk of rejection following pediatric liver transplantation. In this study, we actually asked the most fundamental question using bulk expression signatures, bulk RNA-seq signatures from the peripheral component, peripheral blood mononuclear cells from the circulating compartment. Can we predict whether someone will experience rejection or not? Of course, if you profile expression signatures closer to or at the site of transplant, the signal would be much stronger, but that data is much harder to get. On the other hand, PBMCs, bulk RNA-seq PBMCs is much easier to get, but the signatures are dampened. So the question we asked was, can we, using bulk RNA-seq signatures, predict whether someone would be predisposed to rejection? And we did this both at the pre-transplant as well as at 2 post-transplant time points. And we use 2 distinct approaches. One is the standard gene-centric DEG-based analysis. The other was a module-centric approach where we not just use the gene expression signatures, but we also use the topological structure of the underlying protein-protein interaction network. So, of course, as we all know, that genes are not independent, but the protein products encoded by these genes are part of a complex underlying protein-protein interactome network. So the network-centric approach that we used leverages the structure and topology of the underlying protein network. It clusters the protein network into modules and then overlays expression data onto these modules to use the modules as decision boundaries rather than the genes themselves as decision boundaries. On the expression modules, we of course use a lasso-based approach to classify Rs, patients experiencing rejection, versus NRs, patients who do not experience rejection. This network approach achieves 3 things. First, the signature is dampened because, as I mentioned, this is measured in the peripheral compartment. However, by taking into account the entire module and not just individual genes, we take into account additive effects of genes in the same biological process or pathway or functional module. Second, it does not ignore the topology and structure of the underlying protein-protein interaction network. Third, it achieves dimensionality reduction at 2 levels. First, there is a biologically inspired dimensionality reduction where we are moving from genes and collapsing them to modules. Next is we use L1 regularization to identify a sparse set of predictive module-centric biomarkers. So we compare these 2 approaches. The traditional DEG-based approach, although we identified DEGs, it failed stringent cross-validation and permutation testing, both at the pre as well as the post-transplant time points. But interestingly, the network-based signature did pass stringent cross-validation and permutation testing, and we had moderate predictive power with AUCs between 0.65 to 0.7. However, this is not the final signature, and work is still ongoing to improve this. But this is, to the best of our knowledge, the first ever signature derived from bulk RNA-seq of peripheral blood that can actually discriminate between rejectors and non-rejectors in the context of pediatric liver transplantation. Interestingly, the identified modules that serve as decision boundaries are also, in some cases, targets of existing immunosuppressive drugs. So in this case, I'm showing you the identified network-based signature pre-transplant, and it includes things like type 1 interferon signaling, the complement cascade, cell cycle DNA replication, some of which can be targeted by existing immunosuppressive drugs. This serves two purposes. First, because they are the targets of current immunosuppressive drugs, rather than going through the entire drug discovery or drug repurposing, you could simply titrate the doses of existing drugs or add a third agent. Second, we can do this in a patient-specific manner because we get a per patient per module score, which tells us how likely or predisposed that individual is based on that individual module, and then overall when aggregated across modules. So if you want to know more, please come to both my poster and my oral presentation at ISMB. Thank you very much.